plastic pollution is a massive problem for our environment and ecosystems. Annually, an estimated 8 million metric tons of plastic waste infiltrate in our oceans, driven by inefficient waste management systems and extensive plastic consumption. The fragmentation of plastics, generating smaller secondary particles, poses additional risks, particularly when below 100 micrometers, as aquatic organisms can ingest them. In the same mass of plastic, a higher number of particles are present when the size is smaller. For instance, 1,000 particles of 10 micrometers of polystyrene represent the same mass than one particle of 100 micrometers in size. The reduction of the size results also in a greater area exposed on the surface, which enhances reactivity and interaction potential with surrounding environments. Therefore, having a correlation between concentration, surface area, and number of plastic particles is a key information to understand the potential risk of the small microplastics and nanoparticles. In addition, nanoparticles often exhibit higher chemical reactivity than their larger counterparts. This is due to the increased surface energy and the presence of more active sites on the particle surface. This way, the small size of small microplastic nanoparticles allows them to interact more readily with biological systems that are typically impermeable to larger particles. They can easily cross biological barriers such as cell membranes, the blood-brain barrier, and the placental barrier, leading to potential implications in medicine, but also raising concerns about toxicity and biocompatibility. The fragmentation into nanoscale, particularly below 1 micrometer, transforms the material properties significantly, necessitating a nuanced understanding of their interactions with the environment and living organisms. This transformation demands careful assessment of environmental and health impacts. Then, it is paramount to investigate the amount of small microplastic nanoparticles actually present in the environment, which implies the need for accurate and reliable analysis techniques and processes for them. However, small microplastic nanoparticles cannot be treated as regular microplastics for analysis. Sampling, preconcentration, and further analytical techniques need to be carefully adapted to not lose them in the process. Plastics sized under 10 micrometers cannot be sampled using nets or filter pumps, as done for other sizes. In fact, filters should be avoided and specialized sampling methods must be used to ensure a high recovery and minimize contaminations. Advanced spec concentration and purification techniques to isolate the particles from complex samples must be studied and followed, since acid digestion and the use of hydrogen peroxide are not suitable as they destroy the nanoplastic due to their high surface area. Sophisticated analytic tools, such as a combination of nanoparticles tracking analysis, confocal Raman microscopy, scanning electron microscopy, and pyrolysis gas chromatography mass spectrometry, must be used to accurately characterize their size, morphology, and chemical composition, as other standard techniques such as Fourier transform infrared spectroscopy are not suitable for plastics in this size range. Given this complexity, analyzing the actual occurrence of small microplastic nanoparticles in any given place at any time is virtually impossible due to several significant challenges.